Good morning, everyone. Dr. Page is out of town, so we're going to go ahead and get started. It gives me great pleasure to introduce our speaker, Dr. Tim Kamp. Dr. Kamp did his undergraduate studies at the University of Notre Dame. Then he did his medical degree and a PhD in pharmacology and physiology at the University of Chicago. Then he proceeded to do his residency and fellowship in cardiovascular medicine at Johns Hopkins, and in 1996 he joined our department as an assistant professor of medicine. Dr. Kamp is also the co-founder and director of the Stem Cell and Regenerative Medicine Center, and he's a holder of the Tuchman Chair in Clinical Cardiology. He rose very quickly through the ranks, and in 2008 became a full professor of medicine with an affiliate appointment in physiology. His research has been primarily focused on ion channel proteins and the molecular mechanisms underlying heart failure. In addition, in the last few years, his recent efforts have focused significantly on the application of stem cell research in cardiovascular translational work. His work has been funded by NIH grants and multiple private grants. He has more than 100 peer-reviewed publications. He received multiple awards. I'm going to mention two. The 2009 American Heart Association Top 10 Advances in Cardiovascular Research Award, and more recently, the Rankin Research Award from our own Department of Medicine, which is awarded to a senior member of the medicine faculty who has made a significant research contribution toward advancing the field of medicine. Given his innovative research, he also had multiple patents that have been secured and few patents that are pending. In addition to his research efforts and being a great researcher, Dr. Kamp also is a great clinician and a great educator. He served as the Associate Director for the Medical Scientist Training Program at UW from 1998 to 2007. He participated in multiple medical school classes, including anatomy, physiology, and stem cell biology, and he was involved in the training of many undergraduate students and postdoctoral fellows. I counted 50 since 1997. Dr. Kamp also is a great citizen. He served on multiple peer-review panels, including the AHA National Committee on Cellular Cardiovascular Physiology and Pharmacology, and several NIH study sections. He's on the editorial board of multiple journals, including Circ Research, Stem Cells, and the Journal of Molecular and Cellular Cardiology. He's a reviewer for multiple journals and also is a fellow of the American College of Cardiology, the American Heart Association, the International Society for Stem Cell Research, and the Heart Rhythm Society. So it gives me great pleasure to have him today as our speaker. He's going to be talking about cardiac regeneration, the future. Please join me in welcoming Dr. Kamp.
Thank you so much, Mohammed, for that kind introduction, and thank you, everyone, for being here today. We're going to have some fun today. We're going to talk about the future, so that's always fun because we don't really know what's coming. And is cardiac regeneration part of that future? You'll see that there's hints, but we're still learning. So I will disclose that I'm a consultant for a stem cell company in town called Cellular Dynamics International. So what we'll try to do today is talk to you about what the unmet clinical need is related to heart failure, update you on our understanding of cardiac regenerative biology, where things stand with some clinical trials for cell therapy that have been done both here and around the world, and some thoughts about what the right cells are for the job of cardiac repair, and some of the newer work using induced cardiac progenitor cells and tissue engineering approaches. So a bit to get through, but I think it shouldn't be too bad. So here's the real problem that we're faced in cardiology. Even though our preventive measures have really made a big impact, the rate of death from cardiovascular disease has gone down dramatically in the last 2 decades. What's not going down is the incidence of heart failure. It continues to rise. It's estimated to impact about 6.5 million patients in the United States right now. And according to current predictions, by 2030, it will impact about 8 million. So going up more than 40%. Why is that? Well, as you all know, the population is aging, but there's also increasing prevalence of certain risk factors, including diabetes and obesity. And we're getting better in our care of post-MI patients. So patients are surviving now, but unfortunately some of them are surviving their myocardial infarctions only to go on and suffer from heart failure. Though there's clearly been big advances in our treatment for heart failure, a variety of pharmacologic therapies for heart failure with reduced ejection fraction, including, as you're all aware, beta blockers, ACE inhibitors, angiotensin receptor inhibitors, aldosterone inhibitors, and more recent neprilysin inhibitors. And so we've clearly impacted survival there. Device therapies are helping. Implantable cardiodefibrillators to prevent sudden cardiac death, as well as cardiac resynchronization therapy help as well. And in end-stage cases, ventricular assist devices and certainly heart transplantation is important for end-stage patients. But as we know, the availability of donor organs is limiting how many patients can get that therapy. So with all these therapeutic approaches, we have made progress. It's cost some money, about $30.7 billion, according to the American Heart Association statistics in 2012. And as you can imagine, those numbers are going up over time. But despite all this, the survival after the first diagnosis of heart failure stubbornly remains after 5 years at about 50%. That's a fairly easy number to remember, and unfortunately I've remembered it for a while because we sort of seem stuck there. The landscape of heart failure is changing. We're getting better at our treatment, but more patients are coming with heart failure, and we still have plenty of work to do. So it's in that context that I think some paradigm-shifting therapies are really needed to get us to the next level. And why do I say that? And I think part of that relates to our understanding of cardiac biology, and particularly cardiac regenerative biology. So what I'll tell you on this slide is what I learned in medical school, and that is what happens to the heart after myocardial infarction or other forms of injury. You basically lose the functioning muscle, the cardiac muscle, after a myocardial infarction. This little cross-section of a mouse heart that underwent coronary ligation, and this is trichrome gain, so this is all fibrous tissue compared to the brown-red muscle here. So the idea is that once you lose this functioning muscle, you're stuck with scar. So the more muscle you lose, the more trouble you're in, and there's really no way to get new muscle back, whether it's after a heart attack or if you have myocarditis or other degenerative diseases of the myocardium. And so that, obviously, you can give whatever drugs you want, but the thinking is it's hard to get better than where you are here. You don't want things to get worse, but you're limited by your response. But fortunately, we're learning that it's not quite that bad, because when people actually started looking a little bit more closely, they found out things aren't really quite adding up that way, that maybe there can be some new myocytes. For example, if you painstakingly count the number of myocytes in a rat heart, there are about 23 million in an adult rat heart. And then if you take advantage of markers that tell you what cells are undergoing programmed cell death or apoptosis, you see about 1.3 million per month are undergoing programmed cell death. Yet at 20 months, you still have a heart, and there are about 19 million myocytes. So something has to be counterbalancing this to some extent. And studies also provided some evidence that maybe after injury there's some evidence of cell division in the myocardium. And we also did some accidental experiments, and this is an experiment with heart transplantation, and that is if you transplant a female heart, a female donor, into a male recipient, and you look, if unfortunately the patient succumbs after the transplant, by, in this case, a couple of years, and you do fancy labeling to look for Y chromosomes, that you find in some of the cardiomyocytes Y chromosomes, which you really shouldn't see in a female heart. And it suggests that some male cells have gotten into that heart and somehow formed cardiomyocytes. So maybe, just maybe, there might be new heart cells that can form in a heart. But here's another accidental experiment. I'm sure this isn't why this happened, but during the fun of the Cold War years, when nuclear weapons were first being tested, they were tested above ground, and there were big plumes of radioactive material, including C-14. And so this gave the atmosphere a pulse of C14, so levels of radioactive isotopes went up dramatically. They got throughout the food system, and then in 1963, there was the above-ground nuclear testing ban. So this pulse stopped. So what that resulted in is an environmental sort of blip in the amount of C14 in the environment that got into man, and then because we had the above-ground testing ban, that amount of C14 gradually came down in an exponential fashion. So the idea is that if you take hearts from patients who were alive during that pulse of C14 into the atmosphere, and then you, at autopsy, can take their heart, get the nuclei from the heart cells, and basically look at their DNA content. If it's the same DNA, the same cells, no new cardiomyocytes from when they were born, they should have the same amount of C14 in there as their birth year. It turns out that if you compare the colored lines and the dots, that there's a little dilution for all of these. And you can match them up, blue to blue, a lighter greenish blue to here. You can see there's a little lag, suggesting there's some dilution of the C14 in the DNA. If you do the math, which we're not going to do, but if you look at the paper, if you want to see how it was done, these authors calculated there's about 1% new cardiomyocytes formed per year at age 20. Like many things, it declines as we get a little older to about 0.4% by the age of 75. But by the time you're 50, about 55% of the cardiomyocytes in your heart remain from birth, and about 45% have been generated afterwards. So maybe a hint of good news. There is the possibility that there are new cardiomyocytes that form in the heart over time. It turns out that's the case for man, but we also have some good news from other species that the biology may be more robust at forming cardiomyocytes. And here's some important work using a zebrafish model. And in zebrafish, what Ken Poss did, who's at Duke now, is he cut the tip off a zebrafish heart. And what he found is a clot formed, so the fish survived. But when he did that, after about 60 days, the heart completely regenerated. So here's a species that if you remove a piece of heart muscle, the heart completely regrows. Now, we know that doesn't happen in man. We're nowhere close to that. So a dramatic difference in the zebrafish. And in mammals as a whole, this didn't seem to be the case. So certain amphibia, certain fish will do this. But then recently, and I should also point out that more recently this has been studied by someone named Jensu Kong, who is a new faculty member here at UW in cell and regenerative biology, who has described the genetic control that has enabled this. So I think you're going to see more of that, and hopefully coming from right here. Another story along these lines about hearts repairing and regenerating is we knew the adult mouse heart, adult dog, human heart, if it's injured, primarily scars. But what these investigators did is they said, well, let's check early on. Let's check one day after birth in a mouse and do the zebrafish experiment. Cut off the tip of the heart and see what happens. And what they saw is they could get a clot that could form. So fortunately, the animal didn't exsanguinate. But over time, if you did this experiment very early on in life, you could still get a completely normal heart by 21 days in the mouse. So it turns out, without going into a lot of the details, that the very newborn mouse heart is able to robustly regenerate as well. So again, there must be an important switch during development that occurs that turns off that ability to repair and regenerate myocardium. And there are anecdotal studies in humans, in newborns, that also suggest there may be more robust repair early on in life. And also we're fortunate because one of the leaders in that field is Ahmed Mahmoud, who is also a faculty member here now at UW and is continuing some of this mouse work. So we'll look forward to more there. So how does that happen? How do we get that repair? And can't we do some of that for people? And so there's theoretically a number of different ways that can happen. We can turn on the cell cycle in the cardiomyocytes, get them to proliferate again, which normally they stop proliferating after birth. Maybe the cells have to do what's called dedifferentiate, stop being cardiomyocytes and turn into a precursor cell and expand and then differentiate. Or maybe there are pools of cardiac progenitor cells present in the heart that can turn on and repair the heart that just need to be revved up. So that's gotten people excited, and because, as we know, many of the most common forms of heart disease are due to cell loss, people have thought about trying stem cells from just about everywhere, and this is a figure that highlights some of the different sources, whether they're from blood or bone marrow or skeletal myoblasts or even stem cells from fat, even stem cells from the heart. People have rapidly tried to see what they would do if they were transplanted into the heart. Here's an experiment that was done early on with that concept in mind that used bone marrow-derived stem cells. The idea is we all are pretty familiar with stem cells in the bone marrow. They're pretty important to keep the blood system going. And these investigators said, well, they're stem cells. Maybe they'll make new heart cells. And so they took c-KIT-positive, lineage-negative bone marrow stem cells from a green fluorescent protein-labeled mouse, so all the cells in the mouse were GFP-labeled, and they transplanted them into a non-GFP-positive mouse after myocardial infarction, and they saw large areas of GFP-positive cells that co-stained for cardiac proteins, so they concluded that they had large areas of new myocardium generated by bone marrow stem cells in the heart. And this was the Anversa group and Orlik et al. Published in 2001. So it generated a lot of excitement and a lot of interest to move forward. But it also generated some skeptics. And you'll see that within 3 years, two groups came out and weren't able to reproduce that. Nevertheless, clinical trials moved forward with this excitement, taking bone marrow mononuclear cells and transplanting them post-myocardial infarction in man. And I'll just share, there have been many trials, so I'm not going to go through them all, but I'll just share two of the early prominent ones. And this is the Repair AMI trial, largely a German trial, where they took 204 patients after ST elevation myocardial infarction with a somewhat depressed ejection fraction, and they transplanted bone marrow mononuclear cells about 3 to 7 days after the myocardial infarction, followed up at 4 months looking at ejection fraction and at 1 year for clinical endpoints. And their findings were a reduction in the combined endpoint of death, MI, and revascularization. Pretty big difference, 40 out of 103 to 23 out of 101. So encouraging results from that Repair AMI study, and generated a lot of interest and excitement until you turn to the next article in the New England Journal, the same issue, and that was the ASTAMI trial, which was fairly similar. Patients after an anterior MI got angioplasty and got bone marrow mononuclear cells, followed up for LB function by a number of means. And the bottom line is there was no clear difference in functional parameters in this study. So that is a highlight for the conundrum of the field back in 2006, that we're getting mixed messages. Some trials have shown some signal and others have been less productive. Now, over the subsequent decade, many trials have been done, including participation by Dr. Raval and colleagues here. And overall, this is too much for you to see, but this is from a Cochrane review, which is a sequential analysis of trials and meta-analysis approach, where they sequentially add trial by trial, looking to see if there's benefit or harm above these lines and crossing statistics, or whether you're getting towards futility. And according to this analysis, we're getting pretty close to futility in the post-MI trials with bone marrow mononuclear cells if you look at mortality. So as more and more patients have enrolled, we haven't quite gotten the positive signals that we had hoped for. If you look at autologous cell therapy on mortality in heart failure patients, not in the immediate post-MI period, there's a little more hope that we may be, at least at meta-analysis, starting to see some hint for benefit. But again, these are pooled analyses, and no one major large randomized phase 3 trial has yet been done. So suffice it to say, the jury's still out on this. Some have wanted to call the jury back in. Here's a editorial that came out not long ago, less than a year ago, in a prominent journal, Nature Biotechnology, and you can read there, a futile cycle in cell therapy. Should a cell therapy for heart disease with scant evidence of efficacy continue to be tested in humans? I think they've answered the question in their opinion, but you can read the whole article. And so I think the bone marrow mononuclear cells have reached a stage for the post-MI therapy anyway, that there's substantial skepticism in whether they're really going to have a benefit and whether we should continue to invest our resources in that approach. And I think what we've learned, though, has been substantial over that decade, and that we've learned that if you transplant stem cells just because they're called stem cells and put them in the heart, doesn't mean they're going to form new cardiomyocytes and new cardiac tissue. That was the dream, but the dream didn't quite come straight through. It turns out that the effects, although variable, there are clear benefits, and it's more likely related to secreted factors, whether soluble or in exosomes, whether it's causing cell fusion, reactivating cell cycle, promoting favorable remodeling, inhibiting apoptosis, modulating the immune system, forming new blood vessels, and so forth. There are many different ways that the different cell preparations, when you drill down on the mechanisms, may have some favorable effects. But certainly the simplistic model has not been proven out at this stage. So it's obviously made investigators think, why haven't things been better? Maybe we didn't do enough preclinical studies really to get things moving forward adequately. We haven't figured out, maybe we haven't figured out the cell delivery method and timing appropriately, or dosing and redosing is an important avenue to consider. If the cells just squirt out of the heart, that's probably not a good thing, so we need to think about how to engraft them. Maybe we haven't really figured out the patient population that will benefit most from this, or perhaps we're using the wrong cells for the job. So I want to talk to you a little bit about the latter point, whether we can get a better cell source to repair the heart. And with that, I'll just try and get you up to speed quickly on what I mean by human pluripotent stem cells. The first variety of human pluripotent stem cells are human embryonic stem cells. Those were first derived within walking distance of where you are right now from surplus in vitro fertilization embryos by Jamie Thompson and colleagues in 1998. So 20th anniversary this year, and they took these inner cell mass cells and with informed consent were able to get those and plate them under conditions that they could grow in culture indefinitely, so they self-renew, and they're also pluripotent. They can form all the different cell types present in the body. And taking the knowledge from these human embryonic stem cells and the gene expression pattern, investigators figured out how to take transgenes, some of these highly expressed transcription factors in pluripotent stem cells, express them in skin cells or blood cells, and reprogram them into something called induced pluripotent stem cells that behave very similarly to embryonic stem cells in that they self-renew indefinitely in culture and that they are pluripotent. So that's a nice thing because that gives us access, for example, to get a skin sample. We can reprogram it to induce pluripotent stem cells that we can bank indefinitely, or we can differentiate those cells to cardiac progenitor cells or cardiomyocytes, smooth muscle cells, endothelial cells, theoretically, make a heart patch or deliver them directly into the heart. So all sorts of sort of nice cartoon schemes you can draw up, but many challenges along the way to get these cell sources appropriately ready for this sort of application. Because you really have to carefully characterize these cell sources to make sure that they are robust, pure, no infectious disease considerations. Oftentimes we have to develop matrices or grafts using tissue engineering approaches, and then test those cells or tissues in vitro in the dish to make sure they behave as appropriate, and then study them in animal models of disease, be sure we can get them to engraft and function there, looking at efficacy and their in vivo function, and hopefully disease regression in animal models and their safety. We don't want tumors, obviously. We want to avoid adverse effects, and ultimately they get to human trials. So you can see it's a bit of a road to go from these pluripotent stem cells to clinical trials. And in the cardiac area, one of the biggest worries we've had are arrhythmias. Obviously, if we put in cells and they stimulate life-threatening arrhythmias, we will not have helped the patient too much. And there are many theoretical reasons that I won't belabor for you that these cells could be pro-arrhythmic. They could interfere with coupling in the heart. They could induce abnormal pacemaker activity. They could interfere with innervation of the heart. A number of reasons, but if you're more a glass half full kind of person, you'll say, well, maybe they'll be antiarrhythmic. They'll improve coupling as new myocardium is formed. They'll reduce electrical heterogeneities. Maybe this is the better resynchronization therapy rather than pacemakers. So there are reasons to think that the cells could also have antiarrhythmic effects as well. So we'll just have to wait and see. But one of the first challenges we had to come across is getting adequate numbers of cells. It's one thing to grow human pluripotent stem cells in a dish, but if you're going to do cell therapy in a human heart that has, depending on how you count, 3 to 4 billion cardiomyocytes, you got to make a lot of cardiomyocytes. And so over the years, what we've gotten progressively better at, Jinhua Zhang in my lab, and also in collaboration with Sean Palachuk in chemical and biological engineering, Lance Lian and his lab, have developed protocols that can now get plates and plates of contracting sheets of cardiomyocytes that you can just see an example here. So we can get now billions of cardiomyocytes that we can use for all sorts of applications, both research applications, but also for potentially for cell therapy. So this is enabled by a number of groups, the ability to get, in this case, 1 billion cryopreserved cardiomyocytes that Chuck Murray and colleagues at the University of Washington transplanted into a macaque monkey model of a myocardial infarction. And the yellow area here you see are the cells that he transplanted, the human ES-derived cardiomyocytes. So this is a large graft, so you can get large grafts of these cells that co-stain with the cardiac marker alpha-actinin. So we think these are cardiomyocytes, so large cardiac grafts, and I'm not showing you all the data that electrically couple to the monkey myocardium. So here's a cell population that gives you viable cardiomyocytes that can get into the myocardium and couple in areas of infarction. Bad news is that there can be some ventricular arrhythmias they observed. So here's ventricular tachycardia in the monkeys. It turned out it was fairly short-lived, transient at the beginning of their study, and over time the ventricular arrhythmias dropped off. So a large animal model, there's hints that we can actually make progress and get large numbers of cardiomyocytes to engraft into the heart and integrate, although the risk of arrhythmias remains. Maybe we have to do better and get other cell types to engraft as well. One that we're also thinking about are cardiac fibroblasts. Cardiac fibroblasts are important because they secrete the extracellular matrix that's essential for cardiac function and that allows the heart to take its structure and shape, also signal to cardiomyocytes, allowing them to hypertrophy or not. And so we have worked to get some cardiac fibroblasts, and like all cell lineages derived from human pluripotent stem cells, they sort of mimic normal development. And so we have to get the cells to do the dance down the various different stages of development, from mesoderm to precardiac mesoderm to what we think is a heart field-specific progenitor, one area of the heart, the second heart field, and we can get these cardiac fibroblasts, we believe, that are important in cardiac biology. There are a number of different sources of cardiac fibroblasts in the developing heart, which I won't bore you with, but we have tried to get these fibroblasts using basic FGF signaling, fibroblast growth factor, and we have found conditions where we can get a large number of these cells shown here on flow cytometry. And the cells look like cardiac fibroblasts from the native heart if you look under the microscope. And like cardiac fibroblasts from the native heart, we can split and passage them over a number of passages, getting hundreds of thousands to millions and billions of these cardiac fibroblasts. They, importantly, secrete extracellular matrix proteins, and this shows us immunolabeling. These are confocal images over here. Green is collagen, red is fibronectin. Probably easiest to look at the cross-section view here, reconstructed. These are the nuclei. There's some collagen on throughout. Fibronectin's more on top. This is from the cardiac fibroblasts we made from the pluripotent stem cells, and this is native heart cardiac fibroblasts. So a similar sort of pattern that's different from the dermal fibroblasts. So we think we have cardiac-specific fibroblasts that are growing. And using technology developed with Eric Schmuck, in Eric Schmuck and Amish Raval's lab, we are able to make matrix patches from this ECM, and you can see that here, a decellularized patch of cardiac extracellular matrix. Why is that important? Because that gives you a way to deliver cells or products to the heart that includes native cardiac type matrix, and so ideally the cells will be happier and grow better in that environment. So one of the other challenges, and this one you'll have fun thinking about, one of the other challenges we have to overcome is the immunology of these cells. And so that is, if we use human pluripotent stem cells, they're not going to be matched identically to the recipient. Or if we used induced pluripotent stem cells, unless we use them from that patient, there's not going to be perfect matching. And so the same challenge that solid organ transplantation has. Although there are ways to engineer the cells, hopefully to overcome some of these barriers, but how do you study that short of putting the cells into patients? So there have been a number of different humanized mouse models. So those are mice that are severely immunocompromised, don't have T cells, B cells, or natural killer cells, the so-called NSG mice, that have been reconstituted with fetal tissue. But Matt Brown in Will Burlingham's lab, in collaboration, also with Petros Anastopoulos, one of our pediatric cardiac surgeons here, developed, I think, a novel and powerful way to look at the immunology of pluripotent stem cell products. And so without using fetal tissue. And so the way this protocol works, and it's not easy, there is an IRB protocol where patients who have ultrasound when they're pregnant and find out that unfortunately their child has severe congenital heart disease, who will need surgery soon after birth, so cyanotic heart disease typically, are consented, and what they donate are their cord blood cells, but also when the patient undergoes surgery, they get thymic fragments, because as you know, when they open the chest in a newborn to do surgery, from the surgeon's perspective, in the way is the thymus, so usually a large amount of the thymus is removed. And so what they designed was a strategy where they can take the thymic fragments and harvest them, freeze them down, and then either reprogram them to iPS cells or take some of the thymic fragments and use those fragments to reconstitute the NSG mouse. The thymus is necessary to educate the developing T cells, and then transplant in cord blood cells from the cord that will give the hematopoietic progenitor cells, so you can get a human immune system with functional T cells from this tissue that does not come from aborted fetuses, and also has the advantage that you can get autologous cells that you can test in this model as well. So it's a bit of a difficult model, right? You have to go from mom all the way to this complex mouse model, but it's an elegant model because it gives us a way to really rigorously test in a humanized mouse model allogeneic versus autologous iPS cell products. So that's the cartoon version. Here's the version where we're looking at this in a little more reality. These are the thymic fragments, and Matt cryopreserves these, but some he can transplant into the kidney capsule, and if you remember your anatomy and have good vision, these develop into thymic organoids showing Hassel's corpuscles where the T cells mature, so that's encouraging. And then if you sample the blood over time in these mice using antibodies to human white blood cells, B cells, T cells, you can see evidence for reconstitution of a human immune system in this mouse model. So these are potentially humanized mice that have circulating white blood cells, T and B cells, important for looking at engraftment and tolerance of potential cell therapies. And so what Matt next did is he took cardiomyocytes from a differentiation, not autologous, allogeneic, and transplanted them into the kidney capsule. And what you see here is imaging of the kidney in situ. And if you look just at the white under the capsule, you'll see it pucker. That's actually a cardiac graft in there that is contracting away in the kidney capsule. I know it's not the heart, but it's as close as we can get in the mouse right now. And so it gives us a way to get living cardiac tissue in this humanized mouse. And you can see with the histology that there are cardiac troponin T positive cardiomyocytes there. We get some infiltration with T and B cells, CD4, CD8, into this area, and we're in the process of studying how the immune system recognizes these grafts and various interventions to improve tolerance of these in this humanized mouse model. So again, it's not an easy road to get some of these cellular products, but we're doing detailed basic research here. So I'll just highlight another even more experimental approach that's being done, and that's reprogramming. So reprogramming is the idea that like iPS cells, where fibroblasts were reprogrammed to a pluripotent stem state, can we reprogram a fibroblast-like cell to a cardiac progenitor cell? In other words, if you can only think about heart like me, you don't care about the other cell lineages. You want to get something that focuses on the heart, and so a cardiac progenitor cell can make all the cell types present in the heart. And so the short story in work done by Pratik Lalit in my lab and many others is that you can take a collection of 5 transcription factors or and a chromatin remodeling factor, MESP1, TBX5, GATA4, NKX 2.5, and BAF60C. There won't be a test on that, but 5 factors important in cardiac development, and that's how they got on sort of the candidate list. And you infect the fibroblasts, and under the right conditions, by inhibiting several pathways, you can get what we think are induced cardiac progenitor cells. And I'll tell you why we think that. We used a reporter that could turn on when you get cardiac progenitors, and so we get YFP positive colonies that develop. And if we stain those with antibodies for various cardiac progenitor factors, NKX 2.5, GATA4, IRX4, we see the cells express those factors consistent with them being progenitors. But importantly, if you think you have a cardiac progenitor, the most convincing demonstration of that is if you differentiate the cells. So if you put the cells in conditions where they can become the cell types present in the heart, do you see heart cells, for example? And so what we found is that if we took the cells and put them in culture, they turned off the reporter saying they're no longer progenitor cells, and under the appropriate conditions we could see these little stripes, so alpha-actinin, those are sarcomeres that form, and a variety of myofilament proteins, as well as some smooth muscle cells and some endothelial cells differentiated in vitro, so that's in a dish. But perhaps the most exciting experiment that Pratik did in collaboration with Karen Downs, an embryologist on campus, was to take these cells, and he said, well, they're cardiac progenitor cells. They can develop into heart during development. And so Karen had a nice model where she could take mouse embryos at about embryonic day 7 before the heart is formed and put them in a little rock bottle and keep them alive in culture. And over the next 2 days, from a cardiac crescent of clearly undifferentiated cells, a beating heart can form. So it's a pretty remarkable time in mouse development and in development. And if we squirt our cells in there, do they turn into heart cells too? And so labeling the cells with green fluorescent protein and injecting a couple hundred of them. Remember, mouse embryos, you can't even see them without a microscope, so you can't put many cells in. And what you can see here is that if you look closely right there, that's the heart beating. This is the head fold. This is the heart that developed in 48 hours. This is sort of going around to the tail in the extraembryonic tissue. And if you look with fluorescence, right in that beating heart area are cells. So that was good. They got to the right neighborhood. So are they really cardiac progenitors becoming cardiomyocytes? So we had to look a little further. And the way we did that was with imaging techniques, multiphoton microscopy, and immunolabeling, where we could see our green cells, and this honeycomb appearance you see is one wall of the linear heart tube, and red is for cardiac actin. The holes are where the nuclei are, and you could see that as we image down, this is an embryo that's intact still, so we couldn't image the whole heart, just the top side, but we could see evidence that some of our cells indeed became cardiomyocytes in there. So again, suggesting that the cells we could generate in a dish from skin cells, from fibroblasts, could become cardiac progenitors that then could differentiate cardiac muscle. Of course, we're interested to know what happens if we put these cells after myocardial infarction in mice, and here is an area labeled with DAPI. That's a nuclear dye where there's been a dense infarction in a mouse heart. And if you look at the red cardiac actin, you can see there's only patchy cardiomyocytes present in this area, and some GFP, so some of our transplanted cells. And if you zoom in where the boxes are shown down here, you can see overlap of GFP with some of the cardiac specific myofilament markers, suggesting that our cells could get in, differentiate after myocardial infarction, and form cardiomyocytes, smooth muscle cells, and endothelial cells, again, consistent with them being progenitors. And in this particular experiment, where our surgeons were pretty vigorous, most of the animals died in control, the iCPCs were able to keep the majority of animals alive. So a first experiment, many more to do to characterize these effects. But the good news is that unlike mouse embryonic stem cells that form teratomas, we put our iCPCs, induced cardiac progenitor cells, in the same assay in the mouse. We don't get tumors. We can see some of the cells stick around and differentiate into smooth muscle cells or endothelial cells, but no tumors. So that's one good news about iCPCs. And another way we tested these cells was to use a decellularized heart. And by that I mean you can take animal hearts, small or large, and again, a technology that Amish Raval and Eric Schmuck are expert on. You can basically dissolve out the cellular components and leave the extracellular matrix. And what we found is that we could decellularize a mouse heart. So here it is with Langendorf retrograde perfusion in the lab. And over time, over 24 hours, you can get rid of all the cells, and you're left with this proteinaceous matrix structure. And what Pratik Lalit and Ruben Alexanian, who may be here today, who's one of our fellows now and was a medicine resident here, did, and this is largely Ruben's work, they took the ICPCs, put them in the decellularized heart. They were labeled with GFP, so they saw the cells survived up to a month in this decellularized heart tissue. But that's one thing to survive. But then when they stained for some of the cardiac markers, smooth muscle markers, endothelial cells, they saw that indeed in this decellularized matrix, the induced cardiac progenitor cells could differentiate and form the key cardiac lineages. And if you did optical mapping, looking at calcium, you could stimulate these decellularized tissues that then got the ICPCs and were differentiated, and you could see calcium transients that D. Lang in Alexei Glukov's lab measured for us. And you could even see propagation of those calcium transients across the tissue. Certainly, this is just the beginning of recellularization, and it wasn't anywhere close to a normal heart or a normal contracting heart, but hints of cells surviving, differentiating, and exerting functions that you might hope cardiac tissue might show. So that's a little summary of our one of our newer tools in cardiac regeneration, induced cardiac progenitor cells. And then I'll just finish up with one last technique that we're working on in collaboration to see if we can figure out better ways to repair the myocardium than are currently available. And that takes advantage of tissue engineering approaches, where we generate a variety of different cell preparations, share them with our colleague Nenad Versak and his lab at Duke, where they will engineer them into patches of cardiac muscle that then can be transplanted at the University of Alabama, Birmingham, into large animal models, pig models of myocardial infarction. And this just gives you an example of the scale. This is a patch that's been developed with 10 to the 7th cardiomyocytes, endothelial cells, and smooth muscles, and delivered to the pig heart. We can see evidence that it is alive and conducting in the pig heart. And this is still a relatively new grant, so I can't tell you we've fixed these pig hearts yet. And there's much work to be done, but we're trying to take advantage of tissue engineering approaches to better treat and repair the injured myocardium as well. Just to give you a hint that it sounds pretty complicated, and will this ever get to people? This is something that isn't actually published until Monday, but I happened to come across it. And it's from Philippe Menachet, who's been a leader in getting human embryonic stem cell cardiac therapies forward in man. And what he has done is really the first human embryonic stem cell derivative transplanted into man for heart disease and ischemic cardiomyopathy. Dr. Menachet is a cardiac surgeon, and what he did was a phase 1 study with his colleagues in France, where they took 6 patients aged 18 to 81 with a reduced ejection fraction who had to undergo bypass surgery, and he developed a fibrin patch that he filled with these cardiac progenitor cells derived from human embryonic stem cells, and then he basically put that fibrin patch over the area of the heart where they took from their imaging, knew that there was not good vascularization, but there were no targets to revascularize, so they put this patch over there, sewed the pericardium over it, and then followed these patients for a mean of 18 months. And it was a phase 1, so it's a safety trial. And the good news is it seemed to be safe. They didn't get tumors, they didn't get arrhythmias, and they might have, the only concern was they saw alloimmunization. It was clinically silent, but with the blood tests, they could get some hint for reactivity to the ES cells in 3 of the 6 patients. Again, not a surprise because these cells aren't immunologically matched to the recipient. But the good news is that they could get high quality, large numbers of cells that they could put in a patient and after myocardial infarction, and hopefully a cell source that's going to make a difference. Because there are only 6 patients and it's a safety study, efficacy will have to wait for another day. but it gives you a hint that more studies are on the way with this cell population. So what have I tried to share with you today? I've tried to bring out the idea that heart failure is a growing problem in the United States, and we still haven't taken care of it as well as we hope to. There's still a 5-year, 50% survival. I've also tried to update you that what we learned in medical school, for many of us, may no longer be current. The heart does have some ability to regenerate. It's not just a post-mitotic terminally differentiated organ, and that there are some new cardiomyocytes that form over life. Obviously, we'd like to ramp up that number. So far, the cardiac cell therapy studies have not been as successful as we'd like. And certainly there are many ongoing studies, and we hope that with some of the newer cell preparations, the direction changes. And I think I've also tried to highlight that human pluripotent stem cells can give us opportunities to get all different types of cardiac cell that may be useful for repair. Also, with iPS cells, there's the potential for autologous sources, and we really have a fair amount of work to do to assess these in preclinical models and also make sure they're safe and tolerated. There are new strategies that continue to emerge, whether they're reprogramming the induced cardiac progenitor cells. I didn't tell you about induced cardiomyocytes that other groups have come up with, but certainly another area. Tissue engineering approaches are only going to grow in application, and ways to stimulate proliferation of cardiomyocytes are coming as well. So cardiac regeneration, I think, is in the future, but the future is still a little fuzzy to me. But I think there's good work to be done, and I think a lot of promise remains. So with that, I'll acknowledge that the work from my lab that I shared was done by many investigators, many good colleagues here on campus, and thank you very much for your attention.
We have a few minutes for questions. Bennett? And Tim, if I may ask you to repeat the question, please.
Tim, that was delightful to hear. It's nice to know the heart is not post-mitotic. Maybe we'll see some of those behind this
That's another lecture, Ben.
So, do you rely on the place where you put the cells to decide the level of differentiation, or is this pre-programmed? Because I can imagine it could be either too undifferentiated or too differentiated. How do you figure out the level of differentiation of the cells to not be getting, to not be too independent?
So Bennett's question is, how do we figure out the level of differentiation of the cells that's optimal to repair the myocardium so they get the job done, but also not so much so that they become tumors? And at this point, in fairness, I would say most of the decisions are empiric in nature, and we hope that the intrinsic signaling environment of the heart, since it can slowly regenerate and make some new myocytes, that there are some pathways that will help direct the cardiomyocytes to differentiate appropriately in that area. But it's a challenging question because, as you know, the heart is dynamic after myocardial infarction, for example, or in heart failure, and so oftentimes we transplant the cells in the border zone around that where there is active turnover of tissue, hoping that helps direct some of this. And ultimately, we're lucky that the heart is not a very tumorigenic environment. The continual contracting activity of the heart, and perhaps because of the low turnover of cells, normally don't see a lot of cardiac tumors. So I think it's overall an environment that maybe favored a little bit to protect us against that. But you're exactly right. Optimizing the differentiation, where the cells go, and how they home to where they need to be and generate new tissue in a controlled manner is something that is going to take continued study and effort. It's also one of the reasons some people are focusing more on tissue engineering. So if you can engineer a piece of heart muscle that's already ready to go, and put it into the myocardium. Maybe that's a better way. Unfortunately, our myocardium isn't plug and play, though. We don't have the USB port that we can just sort of put that new muscle in. So there are challenges with tissue engineering. Yes, Mom.
I have a question. Well, you know, a question because every cell has a sex. So does the sex of the cell or the sex of the recipient figure into any of this?
I'm sure the answer is obviously yes. But how? The answer is less obvious. As you saw one of the slides I gave, even in heart transplantation, sex is not always matched. And people, males with female hearts, females with male hearts, seem to do okay as best our analysis and follow-up can detect. At the level of cell therapy, we know even less. But certainly, you could imagine that that could impact things.
Tim, great talk. I have a question about your thoughts related to immunogenicity of cells and kind of the futuristic approaches that are being kind of batted around in the field of how to get cells that may come from donors into other people, diseases that would seem to make sense for an off-the-shelf approach for that allogeneic transfer or diseases where autologous cell therapy might be questioning on banks and pollination protocols and so on. What are your thoughts about how we're going to disseminate this technology?
Thanks, Amish. So Amish asked about the immunologic challenges with these therapies, allogeneic products, which diseases are appropriate for allogeneic products, which ones aren't, and how are we going to get this therapy in a commercially viable fashion that can get out to patients in need. Obviously, that is an area that many investigators and companies, for that matter, as you know, are looking at. So there are strategies, some led by the Japanese in making large banks of HLA-matched stem cell lines, similar to what we do with organ transplantation, and match the major haplotypes, and there are super donors. It's easy in Japan because of the homogeneity of the population. They calculate with just 90 lines, they'll cover most of their population. The numbers are bigger in the U.S. given the diversity here, but certainly master cell banks that are going to be more tolerated with less immunosuppression because there are no major mismatches for some of the cell products are being considered by multiple entities. As well, you're familiar with advancing technology for gene editing using CRISPR-Cas9, for example. So there are strategies to edit human pluripotent stem cells to change some of their major histocompatibility complex antigens, so there are they can be tolerated without immune rejection. And I won't get into the details, but there are many different approaches that are being tested. Actually, if you want to learn more about it, there's a symposium here in Madison on the 18th of April at BTCI where we'll have some of the world's experts on that presenting. And so there are strategies to genetically modify the ES or iPS cell lines as well. When are allogeneic approaches appropriate? I think we're still learning, too, about actually the science and biology. So if you don't need a new cardiomyocyte and your cell only needs to be there transiently, as we've seen with parenchymal stem cells, and secrete exosomes or other cargo that can really positively impact the myocardium, that may be when allogeneic approaches are most beneficial. But for end-stage heart failure, I'm guessing that's probably not going to be the case. But you can have these discussions across the whole spectrum of cardiac diseases that you may consider with cell therapy and a variety of different approaches. So no easy answer on this, sorry.
We're going to end with this question. Tim, thank you for the excellent talk.